Okay, so welcome back to this next video in which we are discussing chronic gastritis and peptic ulcers. Okay, right. What we now want to turn our attention to is the regulation of the secretion of hydrochloric acid by parietal cells. Okay, so basically the first thing that we need to talk about is a type of cell known as the enterochromaffin-like cells. Okay, because these enterochromaffin-like cells, these control how much hydrochloric acid the parietal cells secrete. Okay, now enterochromaffin-like cells are often abbreviated for short to ECL cells. So E is for entero, C is for chromaffin, L is for like, so it's ECL cells. Okay, so let me show these. So basically the enterochromaffin-like cells are located within the lamina propria, okay, and they're located just underneath the parietal cell. So if we go back to this picture, here is our parietal cell here. The enterochromaffin-like cells will be positioned just underneath the basement membrane in the lamina propria, so here, okay, and basically they secrete histamine, which controls the secretion of hydrochloric acid by the parietal cells. So let me draw this here. So let's have our parietal cell here with this funny shape now where we have these canaliculi, okay, and it will then be sitting on a basement membrane. So let me put in the basement membrane in turquoise here, and then underneath the basement membrane we'll then have this special type of cell known as an enterochromaffin-like cell here. And I don't want to make the nucleus of the enterochromaffin-like cell too much bigger than the nucleus that I've drawn for the parietal cell. And basically, these enterochromaffin-like cells secrete a basal level of histamine, okay? So they are always secreting a little bit of histamine onto the parietal cells, okay? And this histamine is what then stimulates the parietal cells to always be releasing a little bit of hydrochloric acid, okay? So the amount of histamine you release is proportional to the amount of hydrochloric acid that the parietal cells release, okay? So the amount of histamine that these parietal cells are receiving from the enterochromaffin-like cells determines their secretion of hydrochloric acid. Now this is an example of paracrine signaling. So paracrine means when one cell secretes a little molecule, a signaling molecule of some sort, which then diffuses out and acts on cells that are nearby it. Okay, so this is an example of paracrine signaling because the enterochromaffin-like cells are secreting histamine, the signaling molecule, and it's acting on cells that are very nearby the enterochromaffin-like cells. Okay, and this histamine will act on H2 receptors, which are on the surface of the parietal cell. Now, H2 receptors are G protein coupled receptors. Okay, so they have these 7 membrane-spanning alpha helices, like so, and then they have a rather small amino-terminal domain on the extracellular aspect of the cell membrane, and then a small carboxylic acid terminal domain on the cytosolic side of the membrane. Okay, and this is an H2 receptor. Now, H2 receptors are in the family of rhodopsin-like G protein-coupled receptors. So G protein-coupled receptors themselves, GPCRs, are a massive great family of proteins. In the human alone, there are 800 different G protein-coupled receptors that are known. Okay, now to give us some sort of way of coping with that enormous number, basically we group G protein-coupled receptors into 5 families. Okay, so the main family of G protein coupled receptors is the rhodopsin family of G protein coupled receptors. Okay, and the H2 receptor is a member of this rhodopsin family of G protein coupled receptors. But there are other families of G protein coupled receptors. So, for instance, another major family of G protein coupled receptors is the secretin family of G protein coupled receptors. Okay, And the secretin family of G protein coupled receptors contains, for instance, the receptors to calcitonin, the receptors to parathyroid hormone, the receptors to glucagon are all in the secretin family of G protein coupled receptors. Okay, next up we've got the glutamate family of G protein coupled receptors. And this basically contains the metabotropic glutamate receptors and the GABA B receptors. Then we've got the adhesion family of the G protein coupled receptors, and this contains a bunch of receptors which bind to components of the extracellular matrix. And then finally, we have a family that is named after its two major members, which is the frizzled taste 2 family, okay, and this contains the frizzled receptor and the taste 2 receptor, okay, and the frizzled receptor is involved in the Wnt beta-catenin pathway, and the taste 2 receptor is involved in the gustatory system. It's involved in sensing bitter tasting molecules. Okay, so after that little sort of diversion, basically the rhodopsin family of G protein coupled receptors is easily the most important. It contains 750 of the known 800 G protein coupled receptors, so it's easily the biggest. And basically the characteristic feature that all rhodopsin family G protein coupled receptors share is that the ligand binds to the G protein coupled receptor on the transmembrane domains, okay, so it binds to residues which are in these 7 membrane spanning alpha helices, okay, so it doesn't bind to residues within the amino terminus, it binds specifically to residues within residues within the 7 membrane spanning alpha helices. And although I've drawn it sort of bound to the 3rd and 4th membrane spanning alpha helix, in reality it's just that it's bound to residues within the 7 membrane spanning alpha helices. Okay, so that's what's special about these rhodopsin family G protein coupled receptors. So histamine from the enterochromaffin-like cells will be binding to these H2 receptors on the surface of the parietal cell, and when the histamine binds, what it will do is it will trigger some sort of conformational change in the heterotrimeric G protein, which will then trigger a change in these intracellular loops here. So we have 3 intracellular loops, 1, 2, 3, labeled intracellular loop 1, intracellular loop 2, intracellular loop 3, or ICL is short for intracellular loop. So this is ICL1, ICL2, and ICL3. And basically these intracellular loops make up a domain which binds to heterotrimeric G proteins, okay? Now, the H2 receptor will specifically act on GS heterotrimeric G proteins, okay, which means that the alpha subunit of this heterotrimeric G protein is a G alpha S subunit. Okay, so the activated H2 receptor will activate the heterotrimeric GSG protein, and the activated GS heterotrimeric G protein, specifically the activated alpha S subunit, will then go on to activate adenylyl cyclase enzymes, which will cause the production of cyclic AMP. That cyclic AMP will activate protein kinase A enzymes, and protein kinase A enzymes will then result in the phosphorylation of the proton potassium ATPase, which then activates it. And of course, we've seen that this proton potassium ATPase is fundamentally what drives the secretion of hydrochloric acid. So that's how histamine activates the secretion of hydrochloric acid, and how much histamine you secrete onto the parietal cells determines how much hydrochloric acid the parietal cells secrete. So, the enterochromaffin-like cells are continuously secreting a little bit of histamine onto these parietal cells, which tells these parietal cells to secrete a continuous sort of little amount of hydrochloric acid, which keeps the stomach nicely acidic. Okay, now, basically, you can increase the level of hydrochloric acid secretion by the parietal cells by increasing the amount of histamine that the enterochromaffin-like cells secrete onto the parietal cells. And this is what gastrin is going to do. Okay, so let's now talk about G cells and gastrin. Okay, so here's a new piece of paper. So, basically, if you eat a very proteinaceous meal, okay, you are going to want a greater secretion of hydrochloric acid so that more digestion can occur. Okay, and that's an awful picture of the stomach, but never mind. Now, basically, there are cells in the antral portion of the stomach, which we've already discussed is this portion down here, okay, which can detect the protein in this meal that you have taken and will respond to it to increase the secretion of hydrochloric acid. Okay, so basically, protein within the lumen of the stomach will make its way down into the gastric pits, down into the gastric glands, and will activate the G cells, okay, which remember stood for gastrin cells, that are at the basis of the gastric glands within the antral wall of the stomach, basically. And these G or gastrin cells will then start secreting gastrin. Okay, so basically the G cells in the wall of the antral portion of the stomach will secrete gastrin, the hormone after which they are named, in response to protein being present within the lumen of the stomach. Okay, and this gastrin will be secreted into the blood. Okay, and now what happens is the gastrin will then act on the enterochromaffin-like cells, which are controlling the secretion of hydrochloric acid by the parietal cells in the body of the stomach, in the wall of the body of the stomach. Okay, so it will come out, so here's the gastrin, and it's now going to act on these enterochromaffin-like cells, which we've just studied. So I'll draw it again. So here's its nucleus. And basically then you have the parietal cell very close by to it. Okay, and we know that the enterochromaffin cell is releasing a basal level of histamine, which tells these parietal cells to secrete a basal level of hydrochloric acid. Basically, what gastrin is going to do is it's going to scream at these enterochromaffin-like cells to secrete more histamine so that the parietal cells will then secrete more hydrochloric acid. Okay, now gastrin works on a G protein coupled receptor that is again in the rhodopsin family of G protein coupled receptors, which is on the surface of the enterochromaffin-like cells. And this is basically the cholecystokinin type 2 receptor, the CCK type 2 receptor. So CCK is short for cholecystokinin, which is another important gut hormone. Okay, so the first C is for chole, the second C is for cysto, and then the K is for kinin. And this is the cholecystokinin type 2 receptor, which again is a rhodopsin family G protein coupled receptor. So the gastrin will bind to the cholecystokinin type 2 receptor somewhere within the transmembrane domain. That will trigger a signaling cascade. And what does this signaling cascade overall lead to? It leads to an increase in the secretion of histamine from these enterochromaffin-like cells. So histamine secretion goes up, and I'll just label this up as an enterochromaffin-like cell. And what will that histamine then do? Well, it will stimulate the H2 receptors on the surface of the parietal cell, which will then activate the GS pathway, leading to the activation of protein kinase A in the parietal cell, and those protein kinase A's will phosphorylate and activate the proton potassium ATPases, which then drive an increase in the secretion of hydrochloric acid from the parietal cells. Okay, so you're going to get more acid being secreted by the parietal cells. Okay, so that's one pathway by which you can increase the secretion of hydrochloric acid by the parietal cells in response to a meal, basically. There is another pathway that I want to talk about, okay, and this involves the vagus nerve, basically. Now, there are two vagus nerves. You have one on both sides, and basically they carry postganglionic parasympathetic neurons. Okay, so basically we are going to get innervation of these parietal cells by postganglionic parasympathetic, and then another big word, cholinergic neurons. Okay, so postganglionic parasympathetic cholinergic neurons. And basically, when you eat a meal, these postganglionic parasympathetic cholinergic neurons will become active, and they innervate the parietal cells. Okay, so let's have one here. So this is one of these neurons here, and basically when they fire, what do they secrete onto the parietal cells? They secrete acetylcholine, ACH for short. So in full, this is acetylcholine. Now, the parietal cells have receptors for acetylcholine, and again, what's beautiful about these pathways is that it's always the same type of receptor. Again, you have a rhodopsin family G protein coupled receptor on the surface of the parietal cells, which binds to the acetylcholine. Okay, now this is specifically the M3 muscarinic acetylcholine receptor. Okay, so the acetylcholine will bind to this M3 muscarinic acetylcholine receptor. Again, the characteristic feature of rhodopsin family G protein coupled receptors is that it will bind to the transmembrane domains, okay, and it will trigger some sort of conformational change which will be translated down to the intracellular loops, which will then change conformation, and now they will have a binding domain for the heterotrimeric G protein. Now, which heterotrimeric G protein do M3 receptors activate? Well, it's a GQ heterotrimeric G protein. So the alpha subunit is the G alpha Q subunit. And basically, when this becomes activated, the G alpha Q subunit will go off and activate phospholipase C beta, which will then it will then break down phosphatidylinositol 4,5-bisphosphate molecules within the inner leaflet of the phospholipid bilayer to diacylglycerol molecules and inositol 1,4,5-trisphosphate molecules. Now, inositol 1,4,5-trisphosphate molecules will act on IP3 receptors within the ER membrane and cause the release of calcium from the intracellular stores. This calcium, along with the diacylglycerol molecules, will then activate protein kinase C, and protein kinase C will do the same thing as protein kinase A. It will phosphorylate the proton potassium ATPase proteins and increase the secretion of hydrochloric acid by increasing the activity of that protein, which drives the whole secretion of hydrochloric acid. Okay, so in response to a meal, these neurons innervating the parietal cells will also fire and stimulate the parietal cells in that way. Okay, so those are the factors regulating the release of hydrochloric acid by these parietal cells. Now, there is one more thing that I should mention, which is the involvement of prostaglandin E. Okay, now prostaglandin E has a protective function for the mucosa of the stomach. Okay, and I want to now discuss this with you. So, basically, the cells of the gastric mucosa produce prostaglandin E. Most cells of the gastric mucosa, they all produce prostaglandin E. So it's not just one specific cell type. All the cells of the gastric mucosa, well, most of them anyway. So the gastric mucosa, remember, consists of the epithelial cells and then the cells within the lamina propria. Okay, so these will be secreting prostaglandin E. Okay, and prostaglandin E is going to have a protective function for the mucosa, but we'll see this in the next video.